INSACOG, Indian SARS-CoV-2 Genomics Consortium। ये नाम आपने कई बार सुना होगा न्यूज़ में। INSACOG, जब भी कोरोना वायरस का नाम आता है, आप हमेशा इस नाम को सुनते हैं। So let's see what this body is all about. Welcome to the channel back, and let's start the video. So the full form of INSACOG basically is Indian SARS-CoV-2 Genomics Consortium. It is a multi-agency consortium of genomic sequencing laboratories. So basically अगर एक सिंपल टर्म में कहा जाए तो ये laboratories है, laboratories for genome sequencing, जहाँ पे research होती है for genomes. इसको establish किया गया था गवर्नमेंट ऑफ इंडिया ने 2020 दिसंबर 30 में जॉइंटली ये जो इनिशिएट की गई थी, ये एक इनिशिएटेड बॉडी है बाय हेल्थ मिनिस्ट्री। सो मिनिस्ट्री ऑफ हेल्थ एंड फैमिली अफेयर्स ने इसको स्टार्ट किया था, एंड डिपार्टमेंट ऑफ बायोटेक्नोलॉजी, CSIR, काउंसिल फॉर साइंटिफिक एंड इंडस्ट्रियल रिसर्च, एंड ICMR, ये तीन बॉडीज के साथ ही एस्टेब्लिशमेंट। So far it has 38 laboratories. Initially जब ये स्टार्ट हुई थी तब इसमें 10 laboratories थी only. Now there are 38 laboratories, and again this is just to monitor the genomic variations of SARS-CoV-2. अगर आपको नहीं पता है तो मैं बता दूं, तो SARS-CoV-2 is nothing but the virus which causes coronavirus disease. तो जो डिजीज है उसका नाम है COVID-19, बट जो वायरस है उसका नाम है SARS-CoV-2. INSACOG, it's a multi-laboratory agency, as we already have talked about, and this is facilitated by National Center for Disease Control, NCDC, Delhi Involving Center Surveillance Unit under Integrated Disease Surveillance Program. So under this INSACOG is being facilitated. Let's see that what is the aim of INSACOG. To understand, there are specifically three objectives for this. But the main objective, of course, is to get research on the SARS-CoV virus and its mutations and the resulting variants, the need for in-depth sequencing and the analysis of the genomic data. This is all the reason that the INSACOG was established. The laboratories were established for this. And specifically, there are three things which are to be focused on it. The very first is to ascertain the status of variants of interest, variants of concern, and in the country. So when we talk about VOC, there are a few more terms with it. Let's understand what these are. जितने भी अभी कोरोना वायरस के वेरिएंट्स अभी आए हैं, उनको बेसिकली थ्रेट्स में डिवाइड किया गया है कि उनका जो थ्रेट लेवल है वो क्या है। तो जो सबसे बेसिक जो थ्रेट लेवल है वो आपका है वेरिएंट बीइंग मॉनिटर्ड, VBM। इसमें कोई भी सिग्निफिकेंट इम्पैक्ट नहीं होता है। दूसरा आपका आता है वेरिएंट ऑफ इंटरेस्ट, लाइक यू विल लिसन दिस, VOI। तो इसमें VOI क्या है? बेसिकली इसमें आपका मे एंड मे नॉट बी the serious level. And if we talk about the next one, that is classified as VOC, which is variant of concern. जब वेरिएंट ऑफ कंसर्न्स की बात आती है, यहाँ पे जितनी भी बॉडीज हैं जो एंटीबॉडीज डेवलप कर चुकी हैं, वेदर बाय द प्री इन्फेक्शन और बाय वैक्सीन्स, वो सारे इन्फेक्शंस भी, वो सारी एंटीबॉडीज भी दोबारा इन्फेक्टेड हो सकती हैं। इस थ्रेट ऑफ लेवल को VOC कहते हैं, ये VOC में आता है। तो अगर आप ओमिक्रॉन की बात करें, ओमिक्रॉन इज ऑल्सो वन ऑफ द VOC वेरिएंट, जो कि वेरिएंट ऑफ कंसर्न है, जहाँ की जो एक इन्फेक्टेड बॉडी दोबारा इन्फेक्ट हो सकती है जो कि एंटीबॉडी डेवलप कर चुकी है, और इवन वैक्सीनेटेड बॉडी भी दोबारा से इन्फेक्टेड हो सकती है। सो दैट इज ओमिक्रॉन इज पार्ट ऑफ इट, इवन द डेल्टा इज ऑल्सो पार्ट ऑफ इट ओनली। And at the end, there's one more threat level, and that is variants of high consequence. In this, there is no case so far for this, and this is very severe condition where there are very rare chances of the person to save the life. So these are the threat levels. So if I talk about the first point, it is just to ascertain the status of variants of interest, variants of concern, and other other threat levels actually. Now the next is to establish the sanitary surveillance and surveillance mechanisms for early detection of genomic variants and assist in formulating effective public health response. So again, after having research on the genomic mechanism, the variants, they provide assistance to formulate effective public health response. Third part is to determine the presence of genomic variants in sample collected during the super spreader events in areas and reporting increasing trend of cases or deaths. तो जहाँ भी आप देखोगे कि बहुत की कुछ स्पेसिफिक स्पॉट्स हैं कि यहाँ पर जो है बहुत ज़्यादा फैल रहा है और इस पर्टिकुलर प्लेस में जो है बहुत ज़्यादा फैल रहा है, सो दे हैव टू डू रिसर्च रिपोर्टिंग ऑफ दैट, ऑफ द increasing trend, and they have to see that how it is spreading and what can be the reasons for it. So as per this it works, and these are the three specific work areas that INSACOG is working for. So let's understand that what is the work process of it. For this, we can see here working of INSACOG. The samples collected for different places are sent to the INSACOG lab. So these are steps basically that how it is working. तो पहले क्या होता है तो जितने भी सैंपल्स हैं वो कलेक्ट करके INSACOG labs में सेंड किए जाते हैं. Then labs जो है वो जीनोम सीक्वेंसिंग कंडक्ट करती है. They try to identify the variant कि कौन सा थ्रेट लेवल है किसी पर्टिकुलर वेरिएंट कौन से वेरिएंट है मीन्स, and then the information is passed to health ministry so that they can take the action for it to address the concern. And this is how the mainly अगर हम देखें तो जो work है that is to just understand the severity of the disease so that the actions can be taken as per that. And this is how the INSACOG works. इसके साथ में एक और टर्म आपने सुना होगा बीच में न्यूज में कभी कभी आ जाता है, so we can understand this to that is SCAG. It is a scientific and clinical advisory group and it was established to assist INSACOG. तो जो INSACOG labs है उनको assist करने के लिए एक और group create किया गया था, that is SCAG. The full form is a Scientific and Clinical Advisory Group. The Central Surveillance Unit this conducts the investigation on variants based on the recommendation made by the SCAG. तो जो ये recommendation देता है इसकी recommendations को आगे करके इनकी recommendation को research किया जाता है and then we can accept it. So basically this is how it is supporting it. So it also has something like to research on only regarding the same aim which INSACOG is made for. And this is all about SCAG. Now we are talking about genome sequencing for a long time. Let's see a like quick note on it. So basically what is this? It is just a sets of DNA. Genome is a set of DNA basically, and genomics is the study of structure. This we should know basically. Like, it can also come in the exam. So it is a study of structure and evolution and function of of the genes in these DNAs only. But ये क्या है genome sequencing? It's just nothing that to find the order of DNA. So it is just nothing but finding the order of DNA. And ये किया कैसे जाता है, इसके कुछ steps होते हैं. If it is asked that we can definitely go for it. So this is first is isolation of the DNA and then fragmentation of DNA, site selection using electrophoresis, paired end sequencing, library construction, and genome assembly. So this is how the sequencing is done. So thanks a lot for watching. This is all about the INSACOG and a few more information on that. If you have something more, please let us know. We'll definitely go and prepare a video for it. Thanks a lot, Jai Hind.